Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikash Bhardwaj. In this video, we are going to learn the difference between prokaryotes and eukaryotes. Here, we are going to have classes of Neet, and in the chapter Cell, the Fundamental Unit of Life, today we are going to discuss the difference between prokaryotes and eukaryotes. So through this video you will learn clearly the difference between prokaryotes and eukaryotes. Now here, if you see the term prokaryotes, pro means here primitive. Pro means primitive. Karyotes, this word it comes from karyon. So in the case of prokaryotes, what you are going to see, primitive type of nucleus. Karyon, here it stands for nucleus. So in case of prokaryotic cell, what you are going to see, primitive type of nucleus. Try to see here, this is the structure of a bacteria. In the bacteria, what you are going to see, the genetic material DNA, it is present within the cytoplasm. It is in direct contact of the cytoplasm, and here if you see the DNA, it is necked. Means around the DNA you are not going to see nuclear membrane. Here keep in mind, in the case of prokaryotes, you are not going to see true nucleus. True nucleus is absent. Here we are going to use the term nucleoid. Yes, keep in mind the examples of prokaryotes, bacteria and archaea. So hopefully through this slide it is clear. In the case of prokaryotes, true nucleus is absent. In the case of prokaryotes, what you are going to see, nucleoid. And what are the examples of prokaryotes? Bacteria and archaea. Now let us move. If you see in the case of eukaryotes, here if you see the term eu, okay, eu stands for true. Karyotes, as I have told you, it comes from the word karyon, means nucleus. In case of eukaryotes, what you are going to see, true nucleus. Try to see the structure of any eukaryotic cell. So within the eukaryotic cell, if you take animal cell, if you take plant cell, what you are going to see, membrane bound nucleus. The genetic material, it is surrounded by a nuclear membrane. So here you are going to see true nucleus, membrane bound nucleus. Now here, what are the examples of eukaryotes? What are the examples of eukaryotes? It is simple, protists, animals, fungi, plants, all these are examples of eukaryotes. Keep in mind, in the case of eukaryotes, what you are going to see, true nucleus. Around the nucleus, you are going to see nuclear envelope. And what are the examples of eukaryotes? Protists, fungi, animals and plants. Now let us move forward. If you see in the case of prokaryotes, what you are going to see, generally the unicellular organisms. What are unicellular organisms? Those organisms in which you are going to see single cell. The organisms which are made up of just single cell, they are known as unicellular organisms. Here in the case of eukaryotes, you are going to see both unicellular as well as multicellular organisms. Here in case of eukaryotes, what you are going to see, unicellular as well as multicellular organisms. So in the case of unicellular organisms, you are going to see just single cell. But in the case of multicellular organisms, what you are going to see, multiple cells, they might be organized in form of tissue, organs, organ systems. Okay, so here in the case of eukaryotes, remember you are going to see both unicellular as well as multicellular organism, but in the case of prokaryotes, what you are going to see, unicellular organisms. If you remember, bacteria and archaea, they are the examples of unicellular organisms. So here keep in mind, the bacteria, they are always unicellular. Prokaryotes, they are always unicellular. They are made up of just one cell. Now, if you discuss about cell size, generally the prokaryotes, they are small. If you see the size 1 to 2 micrometer. If you look into the size of bacteria, it is 1 to 2 micrometer. And if you see in the case of eukaryotes, generally the size of animal cell, plant cell, it lies between 10 to 100 micrometer. So if you see, generally the eukaryotic cell, they are bigger than the prokaryotic cell. Now if we move to the next point, that is related to cell division. Now here keep in mind, in the case of prokaryotes, you are going to see very simple type of cell division process, and hopefully you remember, that is binary fission. In case of bacteria, what you are going to see, bacteria, it will divide into two bacteria just by simple binary fission process. But here if you see in the case of eukaryotes, you may see some complex process like mitosis. So here, based on cell division, what you are going to see, in case of prokaryotes, what you are going to see, binary fission process. In the case of eukaryotes, what you are going to see, the mitosis process. Now come to the next point. Here I have told you, in the case of eukaryotes, what you are going to see, a bigger cell size. So when you are going to have something big, okay, so when you are going to have something bigger cell, the cell, it has to manage its all parts. So within the eukaryotic cell, what you are going to see, compartmentalization. Here you are going to see compartmentalization, and you are going to see different compartments, and all the compartments, they are involved in carrying out specific activities, and these compartments, they are known as cell organelles. Now keep in mind, in case of prokaryotes, cell organelles, they are absent. So hopefully you remember what kind of cell organelles are present in eukaryotes, like mitochondria, chloroplast, in the case of plants, lysosomes. Okay, so keep in mind, cell organelles, they are absent in case of prokaryotes, while if you see in eukaryotes, what you are going to see, compartmentalization within the cell, and here you are going to see presence of cell organelles. Yes, we will discuss role of each one of the cell organelles in detail. Now let us move to the next point, very important point, type of ribosomes. Hopefully you know ribosomes, they are involved in translation, protein synthesis process. Now keep in mind, in the case of prokaryotes, what you are going to see, you are going to see 70S type ribosomes. Here try to see, when you are going to isolate ribosomes from the bacteria, generally you do this by centrifugation, and these ribosomes, they are going to sediment, okay, at some specific Swedberg unit. So here in case of prokaryotes, that is 70S. S here stands for Swedberg unit. And also keep in mind, the prokaryotic ribosomes, they are made up of two subunits. One is large subunit, another is small subunit. So here in case of prokaryotes, remember the large subunit is 50S type, while the small subunit is 30S type. Okay, so here keep in mind, in case of prokaryotes, what you are going to see, 70S ribosomes, okay, and that 70S ribosome, is made up of two subunits. One is large subunit. Here large subunit is 50s, while the small subunit is 30s. Now here if you see eukaryotes, in case of eukaryotes, keep in mind you are going to see 80s type ribosomes. Here in case of eukaryotes, what you are going to see 80s type ribosomes, and 80s type ribosomes, they are also going to have two subunits. One is large subunit, another is small subunit. Here the large subunit is 60s, while the small subunit is 40s. So hopefully it is clear in your mind the ribosomes are of different types in case of prokaryotes and eukaryotes. Now let us move to the very important point, transcription and translation process in prokaryotes and eukaryotes. Hopefully you know what is transcription. Transcription is synthesis of RNA from DNA. Okay, you must be knowing the central dogma of biology, flow of information. DNA encodes for RNA and RNA encodes for protein, the mRNA. So here transcription is synthesis of RNA, translation is synthesis of protein. Now here if you see in the case of prokaryotes, what I have told you, in the case of bacteria, you are not going to see membrane bound nucleus. The DNA, it is necked, okay, it is present within the nucleus and it is in direct contact with the cytoplasm. So as this DNA will be used as a template, RNA will be synthesized, okay, by the way of transcription process. So RNA will come out, okay, and then it will be used as a template by the protein synthesis machinery. Here ribosomes, they will pick up the RNA and then they will synthesize the protein. So here transcription process and translation process, they are coupled. Here you are not going to see any boundary because around the DNA you are not going to have nuclear membrane. So here keep in mind, in case of prokaryote, the transcription and translation process, they are coupled process. Now in the case of eukaryotes, you see in the case of eukaryotes, what you are going to see, membrane bound nucleus. Within the nucleus, what is going to happen, transcription process will happen. So wherever there is DNA, at that place transcription process is going to happen. So here what you are going to see, the DNA synthesis, RNA synthesis will happen within the nucleus. Next, this RNA will be transported out, okay, it will come out in the cytoplasm. Within the cytoplasm you are going to have ribosomes, okay. So translation process is going to happen within the cytoplasm. So both of the events, they are occurring in different compartment. So transcription here in case of eukaryotes, it's occurring in nucleus, while the translation, it is occurring within the cytoplasm. So here if you see in case of eukaryotes, the transcription and translation process, they are not coupled. They are not coupled. So these are the basic difference. Yes, there are many more difference, but keep in mind these are the basic difference between prokaryotes and eukaryotes. Now here if you discuss about DNA. So yes, I have told you in case of prokaryotes, what you are going to see, you are going to have DNA which is in direct contact with the cytoplasm. Here you are not going to have any nuclear membrane. Keep in mind, in case of prokaryotes, what you are going to see, circular DNA. Here what you are going to see, circular DNA. Your genomic DNA of bacteria, it is circular. And here I have told you the DNA, it is confined to a region that is known as nucleus. Another important thing, keep in mind, in bacteria you are also going to see small circular DNA. Okay, so One is genomic DNA, okay, it is comparatively bigger in size, and here within the bacteria you are also going to have multiple copies of small circular DNA. These are known as plasmids. So here in the case of prokaryotic cell bacteria, you can see the presence of plasmids, and when we are going to discuss about plasmids, you will learn that these plasmids may carry drug resistance gene. So keep in mind, in case of bacteria, you are going to have extra chromosomal DNA elements which are known as plasmids. But if you see in the case of eukaryotes, in the case of eukaryotes, you are not going to see generally the circular DNA. Here what you are going to see, you are going to see linear DNA, okay, you are going to see linear DNA, and this linear DNA, it will get compacted, and what you are going to see, the presence of chromosomes. Yes, we will discuss how this DNA will be compacted in the case of eukaryotes. So here keep in mind, in case of eukaryotes, what you are going to see, linear DNA, and this linear DNA, it will get compacted in the form of chromosomes. But here also keep in mind that you are not going to see generally the plasmids. Plasmids generally they are absent in eukaryotic cell, okay, but generally the plasmids they are present within the prokaryotic cell. So these are the some of the basic difference between prokaryotes and eukaryotes. Thank you for watching this. Please do like, share the video and subscribe to my channel Deep Learning Biology. This way we are going to learn, we are going to prepare for Neet medical entrance exam. Please keep in mind, you must have clarity. You must have clarity if you want to clear this exam. The concepts, they must be clear. Yes, sometimes when you are going to watch anything for the first time, you may find difficult. Try to prepare your notes, read from the textbooks, you can learn from other resources, and if you wish, you can watch this video again. You can watch this video again. When you will watch this again, okay, then things will start moving in your long-term memory. That is important. If you are going to read something for the first time, remember it will go in your short-term memory, but if you are going to revise, then definitely it will go in your long-term memory. So keep in mind, okay, by doing more revision, you are moving close to the success. That is the mantra, simple. Okay. Another important point, think right, act right. Definitely you will achieve success. Bye bye, take care.